In this video, we are going to talk about biomolecules. I have a whole playlist on biomolecules, but the difference in this one is that we're going to go over all of them, and we're going to go over them in brief, simple terms. So if you need the nitty-gritty details on any of them, I do have videos for that, but I want to give you this one in case either you need a really quick refresher or you don't have to learn about all the really detailed stuff. So there are 4 classes of biomolecules. They are proteins, carbohydrates, nucleic acids, and lipids. They are alike in that they're all important in biological structure and function, and they're also alike because they are polymers. Polymers are repeating units of things that go on and on and on and make chemical bonds with each other again and again and again to make a longer chain out of smaller monomers or single parts. So these monomers are strung together into a polymer by dehydration synthesis. Dehydration means take away water. So you'll notice in this picture, water is coming out. You have an OH on one of these monomers and an H on the other, and they're going to combine and they're going to leave empty spaces on the monomers. In these empty spaces, the monomers can bond together, and this is the synthesis or making things part of it. In hydrolysis, which is when we take apart polymers to make monomers, we're going to add water, and then we're going to get a splitting. So lysis means breakdown. And don't get confused by the fact that water is being made in dehydration synthesis. It sounds like, oh, the next one should be dehydration synthesis because water's disappearing. But the dehydration refers to the parts of water in the monomers coming out, because the monomers making the polymer are what we care about. And this, again, is not going to happen just for 2 of them linking together, but on the order of hundreds or thousands. So let's talk about each of these classes of macromolecules a little bit. So proteins are made of the monomers of amino acid. The polymer of protein is also called a polypeptide, and these are put together by an amide or peptide linkage. But I'm getting ahead of myself. So the reason we call them amino acids is because they have an amino group and a carboxylic acid group. If you need to know these, then great, look at the groups and you will see what they are. They are called functional groups. These are groups you see again and again in biology. If you don't need to know them, the important part that you do need to know is probably the R on my screen. R is what we use in biology and chemistry to say this can be lots of different organic things. So in this case, it can be 20 different things. And if you look up an image of the 20 amino acids, you will see that there's a lot of different things it can be, from charged to polar to nonpolar. There's a lot of diversity, and this is why proteins can do lots of different functions as compared to the other things that don't do as many functions. Proteins can function in immune system, in structure, in muscle movement, as enzymes, which we'll talk about in a minute, and lots of other things. So the peptide or amide linkage looks like this. You probably don't need to memorize the structure of it, but know that proteins are put together, they're amino acids put together by peptide linkages to form proteins. And like everything else, it's going to be made by dehydration synthesis. You have an amino acid, another amino acid, and you link them by a peptide bond via dehydration synthesis, and water comes out. So for proteins, they're kind of complicated because there are many levels of protein structure. Primary structure is the sequence of amino acids, and this can be hundreds of amino acids and probably is. It's not going to be 3 or 4. So that's just the sequence of amino acids covalently bonded together. A covalent bond is a stronger bond because it's the sharing of electrons between atoms. The subsequent parts of these structures, secondary, tertiary, and quaternary, are bonded by intermolecular forces, which are like hydrogen bonds and other things that are weaker than covalent bonds. So in secondary structure, we have alpha helices and beta sheets, and these are basically structures that are made between different amino acids within the chain from hydrogen bonding. In tertiary structure, we are going to take those little shapes and make them into one big shape. So the tertiary structure is the overall 3D structure of this protein. And then quaternary structure happens only if we have multiple polypeptides or multiple chains of amino acids making one big structure. So in this case, we call each of the polypeptides a subunit, and they all make the protein together. So this one here, this example they're giving, I think, is hemoglobin, which is the protein that carries our oxygen. The most important thing that proteins do probably, and the one you'll hear about the most, is that some proteins are enzymes. So almost all enzymes are proteins, but not all proteins are enzymes. When you see an enzyme, it will have a name ending in ACE, ASE. So this is like carbonic anhydrase, or protease, or lipase, or glucose 6-phosphate dehydrogenase. They're all going to end in ACE. And what enzymes do is they speed up reactions. They do this by lowering the activation energy, which is the little energetic push that the reaction needs to go. They don't alter the reactant, they don't alter the product, they only alter the middle by making things easier. The reactants in an enzymatic reaction are called substrates. Denaturation is not just for enzymes, but for all proteins. You're probably going to hear about it most with enzymes, but denaturation is just the breakdown of protein structure. It's not going to break the peptide bonds or primary structure between the amino acids. It's only going to break those weaker interactions in secondary, tertiary, and quaternary structure, which means that the protein, if you picture the protein fully formed as like a slinky, denaturation is going to be, let's flatten out that slinky. It's just going to be a line. It lost its structure. But if you're going to do hydrolysis and break the peptide bonds, that's like taking a knife and cutting up the slinky into little pieces. I hope you see the difference between those. But denaturation can happen under extreme heat. It can happen under extreme pH, so if something's really acidic or really basic, and it can happen with extreme salinity, so like if I really increase or decrease the salt concentration, and salt is sodium chloride, you might hear it like that. For each of these, every protein, including enzymes, they're going to have an optimal for each of these. So some of them work in really high temperatures, like bacteria that live in hot springs and have enzymes inside them. Or some of them are going to work at weird pH values, like our stomach enzymes that live in acid. So it's less about the absolute number, but more about how the number compares to the protein's optimum. So for example, if I have a stomach acid enzyme that likes being in pH 1 and I put it in pH 3, it is going to denature, and it's going to lose its function because structure and function are very related in biology. So moving on to carbohydrates. Carbohydrates, otherwise known as sugars, are polymers of monosaccharides. Saccharide means sugar. The one you probably know the best is glucose. Glucose is used a lot in metabolism, and it's like our main source of energy. There are other sugars that are similarly related, like fructose, galactose. Those are monosaccharides. You don't need to memorize the structures of carbohydrates. What you do need to know is probably if you see a pentagon or a hexagon in biology, it's a sugar. You can combine monosaccharides to make a disaccharide. Disaccharides are like lactose, which is made of glucose and galactose, or sucrose, which is made of glucose and fructose, or you can do maltose, which is glucose and glucose. And it's not important that you know each of those combinations, but if you see the name, you should know this is a monosaccharide, this is a disaccharide, because that's likely going to show up on whatever quiz or test you have. These linkages that fit the monosaccharides together, they are called a glycosidic bond. And you can see the glycosidic bond is also made from dehydration synthesis, just like the peptide bond. These things all work the same. And you don't really have to memorize what it looks like, but the fact that it's called glycosidic bond should give it away that these are for sugars because you hear the prefix glyco a lot with sugars or carbohydrates. Sugars are primarily used for energy storage, and when they're energy storage sugars, they are not just monosaccharides, but they're in polysaccharide, which is the polymer. There are 3 kinds of polysaccharides that you should know. Glycogen is energy storage in animals, starch is energy storage in plants, and cellulose is in cell walls of plants. We as animals do not have cell walls. We don't have cellulose. We also don't have starch. We can eat starch and digest starch. We cannot digest cellulose, even if we eat it. So these polymers, they're all made of glucose, but they have little structural differences that make them different. But if you memorize like what they do and whether they're in plants and animals, that should be enough. Moving on to nucleic acids. Nucleic acids are DNA and RNA. DNA, RNA. Na stands for nucleic acid. So in the nucleic acid is the polymer. The monomers are called nucleotides. So a nucleotide has 3 parts: a phosphate group, a sugar, which in DNA will be deoxyribose, in RNA will be ribose, and a nitrogenous base, which is the A, G, C, or T, adenine, guanine, thymine, or cytosine, that you've probably heard so much about. The plot twist is that in RNA, they don't have thymine, they have uracil. So in RNA, you don't have A, G, C, and T, you have A, G, C, and U. So I should write that on here, U for RNA. And this is only for DNA. So in order to fit nucleotides together, you will link them with a phosphodiester bond. You can remember this because DNA has phosphate in it, right? So you are going to do this by dehydration synthesis exactly like every other polymer. And I should mention that if you reverse any of these diagrams I've drawn for dehydration synthesis, that's hydrolysis. Lysis meaning breaking, hydro meaning adding water. If you do this lots of times, you get a polymer, which is in this case nucleic acid. DNA is a double-stranded nucleic acid. So you have two polymers and they're going to interact via base pairing. The base pairing means that the nitrogenous bases like each other. So A likes T and G likes C, and they're going to interact by hydrogen bonds. So you'll notice that just like proteins, these polymers are put together by covalent bonds, which are stronger. But the secondary structure, where like two of them combine, or in proteins, that was the quaternary structure, but here it's called secondary. It's confusing. But basically, when you have weaker interactions, they are not covalent bonds, they are hydrogen bonds. And when we replicate DNA, we can split it down the middle and break the hydrogen bonds, and we don't break the covalent bonds. Isn't that awesome? And then in RNA, we are single-stranded, not double-stranded. And it's going to be almost exactly the same. The sugar is not deoxyribose, it is ribose, and the bases don't have T, there is U. But the base pairing rules in DNA are important. C goes with G, A goes with T. And then we need to know what the heck do these things do. Because we knew that carbs did energy storage, we knew that proteins did a lot of crazy things, including catalyzing reactions as enzymes. What do DNA and RNA do? You probably know that DNA stores genetic information. DNA is, I like to compare it to the Constitution. It sits in the nucleus, which is part of the cell, like the Constitution sits in the National Archives. And does anybody pick it up and use it when they want to practice law? No, that's not going to happen because we don't want to get a lawyer's coffee to spill on it and then we have no more Constitution. In the same way, in the cell, we don't want the DNA getting out of the nucleus because what happens if it dies? Or if it gets denatured, or if it gets cut up by hydrolysis? We're done. We don't have any more genes. So we are going to make a cheap knockoff copy. We're going to make the copy of the Constitution that is sold in the gift shop, and it doesn't matter if somebody spills coffee on it. We're going to make RNA, and RNA is going to serve as the instruction manual for proteins. So DNA serves as instructions to make RNA, and RNA serves as instructions to make proteins, and proteins do all the heavy lifting in the cell. So that's DNA and RNA, and that little thing I described here is called the central dogma of biology. And we have one more class to do, that is lipids. So lipids are weird. They are not polymers. I have heard of exactly one teacher out of like at least 10 biology teachers that I've been through that called lipids polymers, and she said the monomer is fatty acid. If your teacher is like that, now you know how to think of it. But most people will say lipids are not polymers. The reason why is because you combine fatty acids and glycerol. Glycerol is a 3-carbon molecule, and it has exactly 3 places for fatty acids to bind. So while other polymers can go on forever and ever and ever, hundreds of times, fats cannot. Lipids stop at 3. This is why that the fats that you store energy in, they're called triglycerides, because tri is 3. So you have 3 fatty acids, and they are bonded to glycerol via ester linkages. So that's the bond for fats, is ester linkage. Fats can be saturated or unsaturated. Unsaturated refers to things that have double bonds between the carbons. So this double bond, that doesn't count. Saturated refers to the fatty acids that do not have double bonds. So these guys are going to be more solid. A saturated fat is like butter. An unsaturated fat is more fluid, like oil. And I should mention that the structures of the fatty acids don't exactly have to be like this or like that or like that. They are just any amount of hydrocarbon, which is not to be confused with carbohydrate. Hydrocarbon is just hydrogen and carbon. So this is a triglyceride. We have 3 fatty acids and we have glycerol. and they are linked by ester linkages. These are hydrophobic. What does that mean? Hydro in this case means water. Phobic is a fear. If I'm claustrophobic, I'm afraid of small spaces. If I'm agoraphobic, I'm afraid of going out of the house. If I'm hydrophobic, I'm afraid of water. So these triglycerides do not like being around water. They are hydrophobic, which is different than all of our other stuff. Another term you might hear is polar and nonpolar. Polar things are hydrophilic. Why? Because water is polar. Nonpolar things are hydrophobic because water is polar and they are not the same. So proteins mostly, and then carbs and nucleic acids, they are all polar and they are hydrophilic. These lipids, they are nonpolar. But what happens if we want to make a phospholipid? A phospholipid is where we replace one of these fatty acids with phosphate. And whenever you hear something that has 8 on the end, that has a charge, usually a negative charge. So this phosphate is negatively charged. This makes it hydrophilic because polarity is kind of like, we kind of have a charge. So if you have a charge, you definitely fall in the category of polar as opposed to nonpolar. So this phosphate is hydrophilic. It loves water. The fatty acids, however, still are hydrophobic. So this has two ends. One of them wants to be with water, and one of them doesn't. And what happens with these phospholipids is they actually start to form spontaneously a bilayer. The bilayer is 2 layers, and all the fatty acid tails face on the inside because they don't want to be around water. All the phosphate heads face on the outside because they're hydrophilic and they love water. Because water is what everything living is floating around in, like all of your cells are surrounded by water in your tissues, even if you don't live in the water, anything on the outside of the membrane will be touching water. And that's how you can remember that the hydrophobic things are on the inside and the hydrophilic things are on the outside. There is one more class of lipids to go through. These are called steroids. Steroids have this 4-fused ring structure I've drawn out. They do have like little differences. You don't have to know those. Steroids are usually going to be named pretty intuitive. Cholesterol, aldosterone, testosterone, and other such names. There's one that I can think of that's not named like that. That is estrogen. Estrogen is also a steroid hormone. And yeah, these steroids they're hormones. Cholesterol serves many purposes that you don't have to really know yet, but these other ones are hormones, and they're going to be signals that go through the blood, and then they bind to a receptor, and they tell a cell what to do. So those are the 4 classes of biomolecules, and that should be a lot of stuff that you need to know. Hopefully it has helped you learn about this.